Yeah, I've been thinking about science for quite some time. When I was a little kid, I can fondly remember, being at the beach, growing up in Hawaii, collecting Portuguese man-o-war. What is it about science that speaks to me? In many ways, it speaks to me because I get to be a kid all the time. For the past nearly 60 years, I have yet to grow up. When you think about, most kids love to discover things, and that's what I get to do pretty much on a daily basis. I am Bradley Moore, Distinguished Professor of Oceanography and Pharmaceutical Chemistry at the University of California, San Diego. My father, Richard Moore, was a professor of chemistry at the University of Hawaii, and he studied the chemistry of toxins in the ocean. He really instilled in me that sense of scientific discovery. So I'm now the second doctor in the family, and my daughter, who just defended her PhD very recently, is now the third in this generation, which is pretty darn cool. Now, I feel super fortunate working at Scripps Oceanography because this job has really allowed me to really think about achieving my dreams in science and research, but at the same time being able to live a life that I really cherish, and melding those two things together. And it's really, the ocean is that centerpiece for me. I was born on an island, so I have that strong connection, and we work with it. Now, the building I'm in, Scholander Hall, which has my laboratories, and part of my laboratories actually are in the same laboratory as Jeffrey Brigam's laboratories from many years ago, which is a pretty fun thing. And we're right on the Pacific Ocean. My labs, my office look out at the ocean on a daily basis. I can see the animals and the dolphins swimming by. I can see the sunsets going into the water. And when I get a little bit restless during the day, I can just run out onto the beach, jog on the beach down for a couple of miles, swim around the pier, and I can be gone in my mind for about 40 minutes. And when I really need to get away from people, I go out into the open ocean and I do long distance kayaking for tens of miles. Often I'm the only one out, and it's a very peaceful place for me to be. It's where my mind can often relax the best. I can think about things, I can dream, and I can just let things go and be in that place where it's just you and the water. Dolphins might be swimming next to you, the pelicans might be flying above, and it's just a really wonderful place. And I would hope more people could be able to enjoy that kind of solitude that we have, where we share a planet with 8 billion people. I can so easily get away from all that, and I love it. So it's a great place for me to dream, to work, and be here at Scripps Oceanography. I feel truly blessed.
I have always been really curious about how the natural world around me is working. When I see a bird landing on a specific tree, or I see a butterfly liking a specific flower, I've always been intrigued as to why they pick that exact spot or that relationship. I think that I'm a naturalist at heart. I like to go out into nature, and I think that science gives me an avenue to explore the language of the natural world around us. I am Natalie Grayson. I am a PhD candidate in marine biology in the lab of Bradley Moore at UCSD Scripps Institution of Oceanography. I also grew up with a love of surfing. My dad started to bring me to Tourmaline and La Jolla Shores when I was around 6 years old. And we had a deal that if I caught 3 or more waves by myself, I would get to go and get a burrito. And I loved burritos, and I still do. I don't think he needed to bribe me with the burritos. I loved the way that you feel weightless in the ocean. I loved the feeling of water covering me. I loved feeling a wave passing over me, or even, if I was lucky, getting to harness the energy of a wave with my surfboard and stand up and ride it. So I've always felt like my most comfortable place was in the ocean. There's not many labs that do as high quality science as we do here that are also on the water. Being here in the lab, doing groundbreaking research, but also be seconds away from jumping in the ocean is really unique to Scripps. You guys can't see it, but actually right outside those windows is the beach. There's a great surf spot here at Scripps Pier. My favorite days are when I can get here early and I can set up my experiments and kind of let them run. We have a lot of great instrumentation in our lab that I don't have to have my hands mixing every second, so I set things up and let them go. And then I go and grab my surfboard and I jump in the water and I think about science or not, just jumping in the water, rinsing off the stress of the day. My expertise in the group is I am the DNA whisperer of sorts. My main part is that I extract really high molecular weight DNA. So almost my goal is to try and get complete chromosomes out of these corals. I do a lot of the bioinformatics, so the sequencing and assembly and kind of making sense of the A's, C's, J's, and T's that we see in DNA. And then I can hand those off, the answers that I find, to other researchers in our group that are working with plants and expressing some of those genes. When in high school and even younger, probably, I would come to this campus and have it as my dream. And I'm living that dream, definitely.
Welcome to the Jeffrey B. Graham Perspectives on Ocean Science lecture.
Well, that was fun. Hey, it's so wonderful to see so many people today, and some good friends are here too. So welcome, it's fantastic to be here. And Natalie and I look forward to sharing some of our science with you today. So let's get started on that. We heard that already. All right. So what I want to do to get this conversation started is for us to think about the natural world, and for us to think about how do most organisms speak to each other. Now, using voice, what I'm using right now, is not how most organisms talk to each other, because most organisms don't have a voice of words. Most organisms don't have ears to listen in on that. But every organism, from the smallest organism to the biggest organisms, have one common language that they all use. And that's the communication, the language of chemistry. All organisms have it. And so when we're having this conversation today, let's also be thinking about how those languages take place between all the organisms on this particular slide or anything else you might be thinking of. And so when we think of it, myself as a chemist, I typically think of a molecule or a chemical as a word. And we know this communication. You all know this communication, whether you're a scientist or not. And maybe you're thinking right now, do I have an example? You probably all have different examples you might think of when you think of how do organisms talk to each other using a chemical. We all have probably one we know very well. Just go to your kitchen and you're gonna notice ants. They all follow each other in a line. Why are they in a line? Well, they know that they're going someplace. There's probably food at the end. They ought to be able to get back. So each ant is depositing a pheromone on a line, and they're following each other. That's communication, means food's this way. That happens everywhere on our planet. We study the ocean, and there are many of these kinds of examples as well. Here's a coral. Coral, and you can see all the little baby corals being ejected into the water column there as larvae, and they're gonna go for a swim. Many of you probably are of the age where you've had children, you'd like them to come back home, and so corals want their children to come back home as well. How do they do that? Well, there's bacteria live on the surface of the coral. They make this chemical that's drawn on this slide here. For the chemists out there, there's 4 bromines attached to 4 carbons with a nitrogen in a ring. That very simple chemical is the scent of mom and dad. It's made by a bacterium on the surface of the coral. And so these larvae, when they finish doing what they do when they go for a swim, need to come back home, settle, get a house, then undergo metamorphosis so they become anchored to the sediment and can start up a new family. It's interesting, this chemical for the nerds out there is designed beautifully in evolution. The chemical begins to fall apart. If this chemical is a signal, you don't want that signal to be in the entire ocean on all the time. So it undergoes a reaction that destroys this chemical. So the signal, as it moves off of the coral, dissipates. And so the larvae is finding it where to go, and they know the highest concentration is where mom and dad is. It also tells it to do those other cool things. Now, all of life has special ways, or their own special languages, using the chemistry. We have it as well as humans. We have our special chemicals as well. Hormones might be one of them. Whether you're a bacterium or whatnot, this slide just shows just sort of a few examples. Some of those examples perhaps you had this morning, like caffeine came from a plant. There are also bacteria or a fungi there that makes penicillin. Some of these chemicals we use, not just to learn more about our natural world, but also we use as humans in our society and our health and whatnot. And many of these chemicals that are specially designed by nature do remarkable things, have impacted us as a species on this planet. Now, all these cells that make these chemicals, that's where my lab comes in. For the past 30 years, we've been studying where are chemicals born in a cell? What do they look like? What do those messages look like? How does this language get started in an organism? Now, if you think back to the central dogma of life, all of information is embedded within DNA. That's where it lives. And when DNA is when we're going to read that sequence, and a cell is going to be able to move that information from DNA where it's archived into active information, converts DNA, gets transcribed to RNA, that gets translated to a protein. If that protein has a function of something called an enzyme, those enzymes come together to make those very chemicals I showed you on the previous slide. So there's this wonderful connection between chemistry and DNA. And that's where I've been studying for the past 30 years. We are curious how we could make those connections, how we could not just learn from it, but how we could predict it and be able to apply it in many different ways. So all small molecules are connected to DNA, whether it be cholesterol in us, caffeine you had from coffee this morning. All this information is embedded within DNA. Every organism has a unique set of DNA, and that information is what makes every organism special and unique. We can learn from that and be able to package that information into organisms. Like take this seaweed here. We can take its DNA, we can put it into something like baker's yeast, and turn that organism into an organism that could make a seaweed chemical. We want to study it in the laboratory. Maybe that organism's chemical has a utility to be maybe important and in human health. So we can turn these little microbes then into factories, into our laboratory to be able to do just that. In my lab, we study a variety of different organisms, and because of my background, as you saw in that video, I have this love affair being in the ocean. Most of the systems we study are in the ocean, whether it be bacteria, whether it be seaweeds, different kinds of invertebrates like sponges or corals, plankton, you name it. Each one of these organisms have a story to tell. Evolution has sort of made each of these organisms special in some particular way. So the workflow in our group is to study DNA that Natalie just talked about in her video, be able to isolate that, understand it, be able to use computational tools to be able to appreciate that, and then be able to manipulate it in some way in a system, and then be able to ultimately make materials, whether it be new pharmaceuticals, whether it be new materials for paint, or maybe make something else for some other property. So that's what we're going to talk about today. And some of those experiences, because I've here as a professor joint between oceanography and the pharmacy school in the medical center allows us to take our discoveries from the ocean into human health. And we've worked for many, many years on developing an anti-cancer drug, for instance, from a bacterium that myself and my colleagues at Scripps have been working on for a number of years. And today, that chemical that is made by a bacterium that lives in the sediments in the Bahamas, who doesn't want to be eaten, no one wants to be eaten, and that chemical is now in phase 3 human clinical trials to treat a very aggressive cancer called glioblastoma. This is a cancer of the brain. So that's pretty cool. Thank you. So what I want to do today is share 3 stories with you. I'm going to share 2 of those, and Natalie's going to share the 3rd story. Each of these stories is a little bit different kind of organisms and the stories that they have. One story deals with a material that we're interested in. Another story is with a toxin, and Natalie's going to talk about a pharmaceutical type of a story as well. All right, so let's get started with the first story. And this story starts with a chemical called Xanthommatin. It's a pigment. And this pigment, as you can see from this video here of a squid, is what allows a squid or an octopus to do that cool thing of camouflage. This pigment allows this color changing ability of these remarkable cephalopods to be able to take place. I know when I was a kid, in addition to being in the ocean, I also liked to disappear. Who doesn't? And so the ability to think about how you could make a material that could change color would be pretty cool. Wouldn't you agree?
Yes.
Awesome. So in addition to, squids, this octopus, we've seen it before. Harry's, the museum just right back there is a wonderful display of an octopus as well. We've all seen that take place and been transformed with how this animal is able to change its shape and its coloration. And so we're curious how this takes place. Now, this is not unique to the ocean world. This chemical called Xanthommatin is also present in a bunch of arthropods and so forth. It's used in compound eye vision. It's used in nuptial coloration. Nature uses it as well for camouflage and for just markings on animals to say, stay away from it. This chemical is in the natural world. It's also something that our world has a strong interest in being able to get a hold of this and be able to use it as a material, a natural material that you could use in paints. We have a grant from the Office of the Navy to be able to look at how we could use this material to be able to camouflage drones or other things like that. You can really go wild in your imagination how you might be able to use a color-changing material like this. But there's a problem. Where am I going to get it from? Am I going to go and probably no one cares if I just grind up a trillion fly eyes, but that's going to be difficult to get. So we need a way to be able to get materials like this so we can study them on a level of an industrial level. So let's take a look at that fly where most things are known about how this material is made. And we're going to look at in its DNA and what's been known. So this chemical called Xanthommatin has 2 colors associated with it, depending on whether it is a reduced form or an oxidized form. In one way it's red, in one way it's yellow. And you can go back and forth between those 2. This chemical comes from a common amino acid. It's one of the 20 amino acids that you and I have called tryptophan. And tryptophan is one of those important amino acids that we need to get from our diet. And in a fly, there are 3 genes, therefore 3 enzymes that convert tryptophan into the building block, and 2 of those 2 molecules come together to fuse together to make this pigment compound. Okay, so suffice to say, a fly does it, that's how the fly does it. And it turns out most other arthropods and anthropods do it the same way. So we want to train a bacterium to be able to do this. So I can make a lot of material. And also I can make a living material eventually to be able to have out of this. So how do we do that? So we co-opted genes from bacteria that look like the genes in these animals. And then we took those genes and we engineered a bacterium and turned it into a little cellular factory to make these. There's a lot of science here. This took probably 4 years to do. It trivialized it on this particular slide here, but there's a lot of work that goes in designing how you do these particular experiments. 4 years it took to be able to get to the point where we can now make a bioreactor, grow a bacteria up, and make large quantities of this particular material. This has been a wonderful achievement, and we now have a patent we filed on this material and this process, and there's a lot of hidden gems in this particular experiment, how to do this. Actually, I just got the galley proofs today from the journal that it got published, that's getting published in for Nature Biotechnology just this morning, and it's been fun reading that and seeing how that looks. Thank you. The person who worked on this, Dr. Leah Bushin, is in a photo here. She and a high school student, Kai Moore, happens to be my daughter, 17. We're also having fun with this material. And so in our lab, we can now begin to paint with this. This is one chemical, and under different, we can spray some acid or some base on it, and the chemical changes color on the paper. Pretty darn cool to see how a material like that begins to change. Leah Bushin just finished as a postdoc with my lab about a month ago, and on September 1 started up as an assistant professor at Stanford University, Department of Chemistry, where she's continuing this kind of work of being able to make cell factories from the work that she did in my lab for the past 5 years. So that's one story, being able to look at sort of nature, be inspired by nature, and be able to think about how do I make a material and do some change with it. I want to have a different story, and it connects back to our ability here at Scripps to be able to work in a building that's right on the water with this wonderful bridge into the ocean that allows my students to be able to be in the laboratory, connect into the ocean. We have wonderful dive capabilities here. Our students can drop boats off the end of the pier and go into the ocean and collect their materials and bring those back into the laboratory. This next project sort of is in that vein and speaks with a project that we started about 10 years ago when this was happening. In 2015, the largest recorded bloom was observed on planet Earth at that time. This was a large bloom of an oceanic plankton that happened in the northeast Pacific that happened. This shut down fisheries because this organism made a toxin. That chemical is shown in the middle of this slide here. It's called domoic acid. It's a neurotoxin, hits a receptor in our brain, and doesn't do good things there. If you have a little bit of it, it's called amnesic shellfish poisoning, so you lose memory, short-term memory. If you have too much of it, it can cause death. And that's why most shellfish that you buy are tested for this particular toxin. Now, when this bloom happened in 2015, it shut down fisheries in the United States along our western coast and caused a lot of harm to those industries that rely on fisheries along that coastline. We were curious about when a bloom like this happens, can we make a prediction using our technology if that bloom is going to be toxic or not toxic? Because blooms in the ocean are super important because that's the base of the food chain. Many of you probably have fish routinely or seafood that you enjoy to eat. They rely on these blooms to take place. We just want to know when the blooms turn toxic. Just like you would like to know if a hurricane is going to come into your town or someone else's or somewhere else. You'd like to understand, make those, be able to forecast accurately when that's going to take place. So that's where we started this project about 10 years ago. These blooms used to happen about every 5 to 10 years. Now they happen almost annually. our planet's changing. Regardless of politics, our planet is undergoing changes today that we're observing. And we see these blooms coming down further south. Los Angeles, for the past several years, has had these blooms taking place, and a lot of marine mammals are, having seizures on the beach, which is never any fun to be able to witness. So a number of years ago, we studied one of these bloom-forming organisms called a diatom. And we sequenced its genome. We studied in the laboratory. We could study in the laboratory under conditions. We could grow the organism up in the lab when it produced a toxin or did not produce a toxin. We could see levels of RNA changing during that time. And other conditions of toxin, we identified a number of important signatures that led us into identifying the genes, 4 genes out of about 35,000 genes. And we could then put those genes together and we could confirm exactly what they did in making the toxin. This now opened up a box that was a black box before to exactly how this toxin was being made. And with this knowledge, we took that and went back into the ocean. We can go up the coast to Monterey Bay, where we had a large NOAA-funded program to work with our friends at Monterey Bay, where we could deposit these large-looking, torpedo-looking devices. These are underwater rovers. They can travel for thousands of kilometers. You can set them into the water. They can move above a bloom, below the bloom, on the sides of the bloom, and they're collecting data the whole time. They collect data in water samples. You can see inside the rover, you can see sort of how it's organized, where a water sample will come in and get divided into 2 parts inside. In one part, it collects cells and it preserves those. On the other side, it actually does a little chemical experiment, an experiment called an ELISA experiment, that tells the operator whether the toxin is in the water at that very time or not, and how much is in there. And this allows us to collect a lot of information. So we collect information over multiple seasons, and we've been doing this up and down the coast. So this slide shows some of the other graduate students in the laboratory. And Stephanie on the top there is somewhere in the audience back here, and she's been going on monthly cruises to be able to collect this valuable information. And over that time, she and others have found something quite startling. And just last year, we published a groundbreaking paper, which now allows us to forecast when a bloom is going to take place. Not react to it, but predict it. This sciencey looking slide here is showing a graph where we can measure the levels of one of the RNA or transcripts for one of the key enzymes that makes the toxin, along with some kind of transporter. When we measure that taking place, 1 week later, toxin gets put into the water. So it gives us now a window of 1 week of a prediction of when an event's going to take place. This is the first time that we've been able to do this for any marine toxin in any ocean system. And I think now this is beginning to sort of teach us how we can make these predictions and be able to impact our societies along the coast. So we're super excited about that. All right, those are my 2 stories I'm going to tell. I'm going to let Natalie Grayson tell her story. And here is Natalie.
Brad loves this picture. I think it looks like I'm about to eat the coral, but that's not what I'm here to talk about. Well, I first want to say how special it is, as you guys heard in our introductory video, that I'm actually from San Diego, and I grew up coming to this aquarium. So 2 weeks before I defend my PhD, to get to come back and talk to the general public about the work that I've done while I've been here is just a really unique experience. So thank you guys all for listening, and I'm going to talk to you a little bit about my project. And so my project starts by us going underwater, and we're going to go through some waves and explore a forest. A different type of forest, though. One not made of trees, but of animals, specifically octocoral. So sea fans, sea whips, and soft corals all fall into this group of octocoral. And they have long branches that flow in the currents like palm trees, fronds, and the wind. And octocoral are colonies made up of polyps that have 8 tentacles, and these tentacles reach out and grab debris and other small bits from the water column, and that's how these organisms eat. And my project is studying octocoral because they are really important ecosystem engineers. They provide structures and habitats for fish, and they can live in diverse areas from the tropics to even here in San Diego, we have some octocoral species. And I set out to understand how these organisms survive, specifically the chemistry that they use. You know, they look really fragile, but actually they're so robust, and they're some of the most ancient animals we have. And I want to unlock the secrets and how they do that and the chemistry that they use to do so. And dolphins also seem interested in unlocking some of these chemical secrets. So while octocoral make chemistry to defend themselves from predators or to maybe deter algae or seaweed from growing on them or prevent diseases. So, you know, we have our skin as barrier to prevent disease, but octocoral are in the water column in the ocean, susceptible to lots of different disease threats. They can make chemistry to prevent the infection of that. And it seems like scientists have noticed this behavior that dolphins can rub up against the coral. In theory, when they have a skin infection and they're using some of the chemistry that the coral is making to cure their own skin infections. And so while a lot of organisms make chemistry specialized, like Brad said, specialized chemistry to themselves, you know, in the world, animals and plants, we've all been interacting and we steal and borrow from each other. And while the dolphins might borrow from the coral, we also have a lot that we can borrow from the coral. And so at Scripps Oceanography, researcher Bill Fennicle, who still works in our building today in the 70s and 80s, did a lot of amazing research looking at the specialized chemistry that the coral was producing. And he found a really fun case that a coral in Australia was this red Gorgonian coral, made a molecule that had some anti-aging properties. And actually, Brad and I are the same age, but I've been putting this compound on my face. I'm just kidding. But this molecule made by this coral is actually in a lot of skincare ingredients on the market today. So this is just a really fun example of, you know, the specialized molecule that the coral is making for itself, it's not making this for us, but we can harness some of that and use it for our own benefit. And while this might be kind of a shocking image with these two divers and this mountain of coral in front of them, in this case, this coral grows really quickly. It can regenerate. If you just cut the top of it off, it can grow back. But if you think about other coral and maybe coral that isn't in such a nice water to access, this is not a sustainable way for us to get enough of this compound for industrial scale application. And to highlight that point, I wanted to share another really interesting case. So this is a coral. It's about the size of my pinky, and it makes this molecule of erythrobin. And this molecule is really active anti-cancer properties. And it was discovered at a really similar time, actually, as a widely available, very strong cancer drug that is on the market. It is Taxol. And Taxol is produced by a tree, the Pacific yew tree, and it was initially found in the bark of these trees. And you can imagine, like people won't want me swimming through coral reefs, decimating the reefs of the coral there. Nobody's going to let someone cut down a bunch of these really beautiful old trees to harness the chemicals in their bark. But scientists were clever, and they found that actually in the pine needles of these trees, a really similar molecule to Taxol was being produced. And so that they could harvest the pine needles, not cut down the trees, and then do some of the last steps of synthesizing Taxol in the laboratory. So that was great. They found a more sustainable way to get this drug from a tree. However, if you guys look at this coral, I don't really see pine needles or an alternative to just cutting this whole pinky off, and I don't want to lose my pinky. This coral wants to be here. So a lot of these really active compounds that were found in coral in the 70s and 80s have kind of just been sitting and waiting. We didn't really have any other way to study them or get at this chemistry without decimating reefs. So this is just to prove my point that corals make really interesting chemistry for themselves, but also there's a huge potential for us to use them in cosmetic and pharmaceutical industry. And I think that this field really burst into life in 2022 when a researcher in Bradley Moore's lab found the genes related to this biosynthesis of those chemicals in the coral's genome. And so what I'm showing here is a phylogenetic tree. If you guys think about family trees, it's just a way to think about how related different things are to each other. So really close, in this case, the same colored branches or lines, those are closely related things to each other. If you see a really long line, like if you look at the really long line before the coral burst out and to have a lot of little arms, that means that those have been evolving independently for a very long time. And that's because corals are very ancient animals. They're some of the most ancient animals, actually, been evolving independently for 750 million years and producing really charismatic chemistry that whole time. I've been learning chemistry while I've been in the Moore lab for about 5 years now. I'm not a good chemist. Corals have been doing this for so long. Why would I try and invent something myself that coral have evolved the capacity to do for that long? Notably, a lot of our drugs today come from plants, but plants aren't even as old as corals. They're 400 million years ago, plants evolved and started making their own chemistry. So just to prove the point that corals are a great place for us to look at for new, interesting chemistry. And now, because the work in the Moore lab, we have an idea about the genes related to how this chemistry is made. And that's where my project gets fun, and that's where I start my work. So I'm showing here 6 different coral that live all over the world. So we have this red coral that's in the Mediterranean Sea. We have that white, feathery looking coral that's in Indonesia. 2 of these are in Florida. And actually 2 of them are here in San Diego. One of them lives under Scripps Pier. This is the sea pansy. And that's a really fun little shiitake mushroom-like coral that lives under the pier. And these coral are really distantly related. They've been evolving independently all over the world for a really long time. And if you think about, when plants or things evolve in their own unique habitats, they kind of spread out. They have their own niche space that they evolve into. So it was really curious for me to see that these coral all were making the exact same chemistry as each other. So that, if we think about why I want to study these molecules, I'm interested in them, why the coral are making them for themselves. That's great. And this is a really interesting question. Coral living in all over the world, making the same chemistry as each other. That's very curious. But also, if we're interested in finding the genes that these corals use to make this chemistry, and I want to do some detective work, because finding the genes that make some really elaborate chemistry is not easy to do, especially when you're working in an animal that is a non-model system that people have never really sequenced the genomes of before. We don't know what its genes look like, how it encodes them. We don't know much. It's kind of the Wild West in these coral genomes, I'll tell you guys that. But if I see these coral have diverged so much, and their genetic information, when I look at it and compare it to each other, is really different. There's only a few things that they have that are conserved, and it's some cases the genes related to the chemistry that I'm interested in studying. And so to do that, I need to generate some really great genomic data for these corals, which had never been done before. So my PhD was harnessing the DNA and trying to pull out the secrets that these coral encode in their genomes by doing some genomic sequencing. And so here, this clear liquid that's getting pipetted into this, it's kind of like a big USB port looking like thing, that actually contains DNA, and we're doing this in the lab. And I'm loading the DNA into like a chip-like thing that has all these pores, and the DNA molecules just load themselves into the pores, and we can see green, woohoo, that means we're sequencing DNA. So that's live reading those base pairs, those A, C, Gs, and Ts that I loaded into that chip. And this is really remarkable. This is, being in San Diego, there's a lot of biotech. There's a huge advancements going on in our sequencing capacities. And this step of being able to sequence the DNA of an organism in the lab is amazing because DNA degrades the second that organism dies. So I get to get really high quality DNA out of these corals. But you can imagine something that's that small. I can put that in my pocket and I can go to some remote field site. Instead of having to bring that coral back home with me, I can sequence its DNA on the spot. And that is a really beautiful advancement in our field. So now we can sequence the genomes of these organisms that we really had no previous way of studying. So with this project, I got to do a lot of this great sequencing of DNA, and then that's when the fun really begins, because Brad made an oversimplification of this central dogma. Oh, DNA to RNA to protein to molecule. It's not that simple. It's not just one color. It's, thousands and thousands of genes, especially, okay, you're looking in a bacteria, that's one thing. You're looking in an animal. We have so much DNA, and it's not tightly packaged into little circular pieces. It's, in chromosomes, it's huge, it's really long. And if you're thinking about the biosynthesis of a complex molecule, like I'm showing here, that is kind of a crazy molecule. When I didn't know much about chemistry and I saw something like that, there's no way I'm touching that thing. But, and to make that molecule, it's not just one gene in a genome. You need to pull out, the pink genes, the green genes, the blue genes, and those are transcribed into RNA and translated into unique proteins that come together kind of like, a building block building a house, assembling something. They each have their own role. So maybe blue is going to make one bond and an orange protein is going to attach something else. And they work in this concerted fashion in a really complex way. So when you're thinking about trying to make a molecule that's this complicated, you need to find a lot of genes encoding each of those steps. And so that is exactly what I got to go ahead and do as a detective. And I looked in these coral genomes and I found the genes related in part, I found the genes related to the biosynthesis of this molecule. And I got really lucky, instead of having to find, it's like searching for a needle in a haystack. And if you're looking for pink, orange, those are a bunch of different haystacks because DNA isn't just one pile of hay, it's tons, and I need many, many needles. But I got really lucky that a bunch of the genes related to the biosynthesis of this molecule were all tightly packaged for me in the coral's genome. And so I got to find them across all of those 6 different coral, and they all had the same conserved genes to make this molecule. Okay, great, Natalie. You found the genes that you think are related to this really fancy molecule. How does that solve the problem of us being able to produce this molecule at scale to solve human disease? Well, that's where things get fun. We, in a lab setting, very controlled, get to engineer microbes and yeast to produce, as Brad said, as cell factories to make our chemistry. And we take it one step farther. We actually force a bacteria that is known to be able to hijack plants to make their genes. We hijack the bacteria that's doing the hijacking, actually, because we put coral genes in the bacteria, and that bacteria puts our coral's genes into the plant. And so now we have a plant that's making coral chemistry, which is incredible. And this work, it would not be possible without another graduate student in our lab, Hannah Bone. She is, an incredible scientist, and this is the first time someone has ever put a coral gene into a plant. That is mind-blowing to me. It's one thing to have yeast or bacteria, maybe making some that you can clone them because they're really simple, but a plant is a really complex organism, and we are now using it instead of having to go and kill tons of coral to get at our chemistry. We're growing plants, we're growing, and these plants are making our chemistry for us. So this is just a proof of concept that this field of looking for chemistry in marine organisms that are really sensitive, and maybe there's not that many of them, is more promising, I think, than a lot of us had ever thought. We can tread lightly on these reefs and just get enough to sequence their DNA. And then we can clone and put these genes nicely packaged into these expression systems like bacteria and tobacco. And we can start to make some of this chemistry ourselves at scale for drugs. And I want to leave you guys with just a little bit of what we're doing next. And so because we can tread so lightly, we can actually explore some really exciting horizons. And not just horizons, but maybe below the horizons, the deep sea. And so we got to work with the Jensen lab, which is another lab at Scripps, and we got to use some of these amazing Scripps research vessels to go on an expedition and collect a bunch of coral, small amounts, coral that lived in the deep sea, and sequence their genomes. And we actually found some new species, new to science species of coral living there. And we got to categorize the chemistry that we think they can make because we can sequence their genomes. And so, by using genomics and coupling that with chemistry, we can think about, what are other really sensitive or pristine systems that we don't want to ruin and destroy. And that's not what scientists are here to do. But we can study them and we can harness the chemistry that they've been evolving their whole lives and for millions of years. And we can start to now use it for ourselves. So I'll bring Brad back up to wrap it up for you guys.
All right, so we have a couple slides just to sort of finish things up here. So as Natalie said, we've made a lot of progress over the past number of years in being able to make this wonderful connection of the specialized chemistry of life and bring it back to DNA and then bring it forward to be able to make new materials and so forth. My group has been able to do this in a number of different areas, not just in medicine, but also in food security and in biotechnology. They all have the same types of connections there, but they all require us to be able to learn from nature. And, nature, as our teacher, has a lot to tell us, a lot to teach us. And we need to have our eyes open as we sort of be able to learn from these experiences. But we also need a planet that's going to be able to be safe and be able to be that place where nature is able to continue to flourish and continue to be able to teach us. We happen to be at a moment in our time where species loss is accelerating. We're at the 6 so-called mass extinctions that we're experiencing right now. This is the first time it's been human-caused for these mass extinctions that are taking place. Some people call it an annihilation of the tree of life. Every time we lose a species, we lose a teacher. We lose that information that organism somehow evolved and did something over millions of years and maintained its place on this planet. We lose that knowledge. So our group is really trying to have this race against time in many ways, to be able to sequence as many organisms as we can, to be able to learn from this and apply that knowledge so we could perhaps address future types of challenges that we might have that either we can study in my own lab or other scientists can also take forward as well. But we see wonderful hope, that this technology and this ability of this time and this moment is allowing us to do just that. And although there is a race against time, we feel we're up for that particular challenge. So I hope that we've shared a few stories with you that sort of hit home to some of you, and we'd be happy to answer any questions that you might have after this. I'm just going to end with a slide showing my research laboratory. Our group is full of scientists who are students here at UC San Diego, whether it be undergraduates, graduate students, or postdocs. They are chemists, they're biologists, they are explorers, they work in the field, they work in the laboratory, many different skills. And I've called out a number of them, and these are the names associated with those 3 projects that we shared with you earlier today. One more thing I want to point out on this slide. It doesn't take just a lot of, power of this remarkable team that I've been able to amass here, but also the funders that support us to do this kind of work. Our work comes primarily from the federal government, from the NIH, the National Science Foundation, NOAA, Defense Department. We're also very fortunate to get funding from private philanthropy as well, or foundations like the Moore Foundation, Gates Foundation, and others as well. And so that means we need to thank you. We need to thank you because you're taxpayers. We need to thank you, we need to thank your parents, we need to thank your parents' parents, who for generations have contributed to this wonderful experiment we've had here in the United States of having a remarkable public science capabilities in our universities that have really made our country here the envy of the world. So thank you for making that happen. I hope it's something that we can continue as a society moving forward. Thank you.